Here we will discuss the answer to some of the homework questions, especially those regarding what happens when either you prevent oxygen from binding to the last protein in the electron transport chain, and what happens when you have a leaky membrane in the mitochondrion, and finally the difference between fatty acids and the metabolisms of carbohydrates. If you have a leaky membrane, that is a membrane that has some sort of pore through which protons can escape. And so if you think that the whole purpose of the electron transport chain is to use the energy in those electrons to create a proton gradient, having pores through which those protons can escape means that the gradient will disappear. And if you don't have a proton gradient, if you don't have a high concentration of protons in the intermembrane space, there are no protons to go through ATP synthase, which needs those protons in order to make ATP. So if there is no gradient, if the protons are escaping through another route and not going through ATP synthase, you won't have any ATP synthesis. And this was used as a weight loss drug for some time, and it was quickly discontinued because it was just a slight dose increase in the dosage will result in lethal effect. So people taking this drug were in very high risk of dying for just a slight increase in the dosage. And the reason is there was a medication that was making these proton pores so that the protons in the mitochondria will just escape, and the cell will go through the entire process of cellular respiration but would not be able to make any ATP. It was a weight loss process because it was a weight loss drug because the cell was still doing all the steps in cellular respiration, that is not just glycolysis but also going through pyruvate oxidation, in which you lose some of the carbons as CO2, and then Krebs cycle, where you completely lose all the carbons in the glucose molecules. So in this process, the cell was still converting all the carbons and the glucose into CO2, which is what results in the weight loss. But it was not making any ATP, so this was forcing the cell to do this many times, but not getting the ATP production. So the only ATP that the cell was getting was the ATP from glycolysis, which is 2 ATPs, and not much of the ATP synthesis here, which, as we've seen in the past, this is what provides the main bulk of the ATP production. So this gives you 30-something ATPs compared to just 2 that you get from glycolysis. So at the end, the people who were dying from a lack of ATP. So their effect was pretty much the same as if they weren't doing any cellular respiration, or sustaining with just glycolysis, which is not enough to sustain life. Cyanide poisoning is also deadly, as we know, and it works slightly different. So what cyanide does is it interferes in the interaction between oxygen and the last protein in the electron transport chain. So it prevents oxygen from taking these electrons at the end of the electron transport chain. So there is no water being formed, but more important than that, since the electrons are not transferred, this blocks this protein from taking new electrons from the transport chain. So this essentially stops the movement of electrons through the electron transport chain. If this protein cannot get rid of its used electrons that no longer have energy, then it cannot take new ones that are coming in with energy, and it won't be able to pump a proton. And then this one will have the same problem. It won't be able to give the electrons to the next protein, and it won't be able to receive electrons from the previous protein, so it won't have new electrons to use in order to pump protons. So there is no electron transport. There is a jam of electrons where this electron has nowhere to go, this electron has nowhere to go, this one has nowhere to go. And so you cannot take new electrons from NADH, so NADH cannot be converted back into NAD+, and NADH has no place to take its electrons to. So there is no electron transport, there is a jam. And if there is no electron transport, then there is no energy for these proton pumps to pump protons. So there is no protons being pumped. So at some point the protons that were there would already move back and there would be no proton gradient. If we have no proton gradient, then we have no protons to go through ATP synthase, which means we have no means of producing ATP. So cyanide poisoning has the effect on the cell equal as having no oxygen, because when you have no oxygen, oxygen is the final electron acceptor. If you cannot give the electrons to oxygen, then you cannot move electrons through here. And the main purpose of the part of the cellular respiration that takes place in the mitochondria is to harvest electrons to bring to the electron transport chain. So if the electron transport chain is backed up, then there is no point in producing any more NADH, and the Krebs cycle will stop, because Krebs cycle mostly is the one that produces most of the NADH, and it needs NAD plus in order to produce NADH. So it will run out of NAD plus and the Krebs cycle will stop. Pyruvate oxidation, also the main purpose is to produce NADH and get the Acetyl-CoA so it can go into Krebs cycle. So that will also stop. So the only thing that the cell will be able to do is glycolysis. So the only pathway that will be happening in a cell that has been poisoned with cyanide is glycolysis. But even glycolysis also needs NAD+, because glycolysis is converting two NAD pluses into two NADHs by giving it a pair of electrons from glucose. So if there is no electron transport chain where this NADH can be recycled back into NAD+, the cell will have to do fermentation. And in fermentation, pyruvate is used as the electron acceptor for NADH. So NADH will give its electrons to pyruvate and become NAD plus again so that it can go back to glycolysis. And pyruvate in the process will turn into either lactic acid or ethanol, depending whether a plant or yeast will be ethanol, and if you are a human or animal, you will have lactic acid, and a CO2 molecule for each pyruvate. But notice that in this process, there is no ATP being produced. The only reason for running fermentation is so that glycolysis can continue and you get the two ATPs that you get from glycolysis. But lactic acid fermentation or ethanol fermentation are the last resource for the cell, to the point where lactic acid is it's not a pleasant result. If you exercise for too long, you feel the effect of lactic acid as your muscles get sore. So this is not something that cells want to do. And ethanol is toxic for cells too. This is cells that are like yeast doing fermentation and producing ethanol as a result get to a point where they can no longer survive because the ethanol concentration is too high for them to live. That's why you don't get a beer or wine that is above a certain level of alcohol, because at that point the yeast starts dying from excess of alcohol. So just to summarize, the only reason for doing fermentation is just so that you can restore this NADH back into NAD+, and glycolysis can continue producing its two ATPs. Now let's take a quick look at what happens when our bodies are metabolizing fatty acids. Like we said, glycolysis is the only process that can go on without oxygen. Any other process needs oxygen, and the metabolism of fatty acids is one that requires oxygen. So our body will only burn fatty acids or fat for energy if oxygen is present. That's why aerobic exercise is the only way to burn fat. But let's see why. Why is that the case? Why is it that way? So this is the process of beta oxidation, which is the process of catabolizing fats. And as you can see here, you need ATP, so you put an ATP in to get the process started. So this is kind of similar to glycolysis in that sense. You have to put some energy to get it started. And then you start harvesting electrons, and you also bind to a CoA, because your final product at the end is this Acetyl-CoA, which is the same Acetyl-CoA that you produce at the end of pyruvate oxidation so that it can go into Krebs cycle. And so that's also the point here in this fatty acid oxidation, is to take this Acetyl-CoA into Krebs cycle. So one big difference between fatty acids and why they can produce so much ATP is because every carbon in the fatty acid goes into Krebs cycle. If you remember from glucose, a 6-carbon glucose molecule, only 4 of the carbons go into Krebs cycle. So a 6-carbon glucose molecule runs the Krebs cycle twice. On the other hand, a 6-carbon fatty acid will produce 3 molecules of acetyl-CoA so that it will run the Krebs cycle 3 times. So that means that you will produce a lot more NADH from a fatty acid and 1 more FADH too. And these are important because we know that those molecules can then bring their electrons, the electrons they harvested from the fatty acid in this case, into the electron transport chain, and then by chemiosmosis you produce ATP. So the other thing to note here is you haven't made any ATP in this process. So all we've done is been harvesting electrons. So we harvested here an FADH2, electrons were given there, and electrons were given here to NADH. The only way to harvest the energy from these electron carriers, which at this point all the energy in the fatty acids is in those electron carriers. And if you remember, those come from this high energy bonds. So the CH bonds, those are the bonds where we're stealing the electrons from and giving them to FADH. So all the energy in the fatty acid molecule is now in these electron carriers that are carrying high energy electrons taken from these CH bonds. And to get the energy from those electrons, we have to do electron transport chain and chemiosmosis. And like we've seen in previous videos, you need you need oxygen in order to run the electron transport chain, because oxygen is the final electron acceptor at the end of the electron transport chain, and without oxygen there will be a traffic jam of electrons. So the electron transport chain without oxygen cannot run, and if you don't have oxygen you won't be able to produce the proton gradient that then drives chemiosmosis to produce ATP. And so all this process of metabolizing fats for energy can only occur in the presence of oxygen.